So what about lipid metabolism? Well, lipid metabolism is a little bit more complex. Lipid metabolism is going to involve the modification or the conversion of Krebs cycle intermediates and small chain fatty acids into triglycerides eventually. Not only triglycerides, but other types of fats, phospholipids, or cholesterol molecules. Lipids are important and are necessary for various physiological functions. Membrane assemblage, the production of steroid hormones from the cholesterol molecule, neuron functions, in particular the development of myelin, vitamin depositing and transportation, in particular the lipid soluble, body ornamentation, that is the depositing of fat in distinct regions of the body. Body ornamentation is particularly important for attraction of the mate, as well as for protection. Regardless of what you might have been told, the amount of lipogenesis that is taking place is not about the quote-unquote caloric balance, not about having too much energy being loaded as opposed to being expended. The rate and the amount is based off of the amount of metabolites, glucose and fructose metabolites in particular, and free fatty acids within the tissues of the body, secondary to hormone signals related to the caloric balance. The glucose and the fructose metabolites that are of importance here are those metabolites that we will see within the Krebs cycle, where when we start getting a backlog of Krebs cycle intermediates, we have an intracellular signal that says time to turn the excess of sugars into carbohydrates. So what is lipogenesis? Lipogenesis, in this case here, is the formation of the triglyceride, the TG, from a polycarbon chain and from carbohydrate, in particular fructose and glucose metabolites. When we're talking about the polycarbon chain, what we're basically talking about is a short chain fatty acid or a fatty acid that can be elongated into one of the precursors in triglyceride formations. The majority of lipogenesis is going to occur within the liver, within adipose tissue, and this is either going to be subcutaneous adipose tissue or the ornamental fat, or visceral adipose tissue, that's the adipose tissue that we find in the mesenteries and surrounding the internal organs, within skeletal muscle, within cardiac muscle, and within connective tissue. The connective tissue fat, or the fat pads, is different than the visceral fat that we see in the mesenchyme connective tissue. They're two distinct things. The visceral fat is the fat that is sometimes the culprit for many of the health issues that we see with overfatness. So lipogenesis is a highly regulated pathway. It occurs under hormone regulations that will either increase or decrease lipogenesis. The hormones include insulin, cortisol, estrogen, growth hormone, testosterone, thyroid hormones, as well as adipokines such as adipocin, leptin, and adiponectin, and myokines, especially irisin, and a myokine-derived interleukin number 6. Along with the hormone regulation, we have regulation within the cell based off of Krebs cycle intermediates, in particular increasing citrate, malate, and oxaloacetate, OAA, concentrations within the mitochondria. Now, what we're going to get into is the biochemical pathways here in a second. The biochemical pathways are going to be predominantly found centered in the mitochondria with some shuttling into the cytosol. Lipid genesis and lipid production is a mitochondrial regulated pathway. And that's key for the production of lipid hormones, steroids, as well as short chain and long chain fatty acids. Just like beta oxidation is occurring within the mitochondria, lipogenesis will also be occurring within the mitochondria, with shuttling between the mitochondria and the cytosol itself. So here we have an overview of the lipogenic biochemical pathway that's going to ultimately lead to the formation of the triglyceride, or the lipid droplet, within cells and tissues of the body. In this, the key regulator is the development of intermediates between glycerol 3-phosphate and phosphophthalic acid. These intermediates are dependent upon various acetylations and transformations of either short chain fatty acids, that's the plus R1 that you see coming from the side chain, or the increase of glycerol 3-phosphate within the cytosol. So let's take a look at this in terms of a schematic, looking at the formation of the triglyceride in terms of regulation. So we have excessive amounts of citrate. Excessive amounts of citrate develop a metabolite-dependent pathway in which citrate is going to be transported out of the mitochondria. It's going to interact with an enzyme that is an ATP citrate-talase, which is going to split the citrate into its two components, OAA and acetyl-CoA. The acetyl-CoA will form malonyl-CoA, which we saw in a couple other pathways, via alkoxylation. OAA will regrade back to malate, malate back to pyruvate, and pyruvate back to acetyl-CoA, which will form more citrate with a interaction with OAA within the mitochondria, and the cycle continues. The cycle continues as long as we have high amounts of citrate. This occurs when the mitochondria continues to do Krebs, but is not sending or not receiving a signal for the need for additional ATP. We have additional things going on here, such as hormone signaling, which is going to stimulate a hormone-sensitive fatty acid synthase, as well as uptake by fatty acid binding proteins of short-chain fatty acids. Most of these short-chain fatty acids are going to be stripped from the VLDL molecules. The short-chain fatty acid will either be shuttled in and cleaved into acetyl-CoA, which will then form malonyl-CoA, or will get interacted with the fatty acid synthase to form palmitate. Palmitate is the first fatty acid chain which will develop into the fatty acid. Coming from glycolysis, because once again we don't need the ATP, we have the glycerol aldehyde 3-phosphate molecule. This glyceraldehyde 3-phosphate molecule has its phosphate stripped and the glyceraldehyde converted to glycerol. The palmitate will undergo an elongation sequence, forming stearate. Via an acetyltransferase, palmitate and stearate can start linking onto the glycerol, and they link on one at a time from the first carbon through the third carbon in glycerol, first forming a monoglyceride, then forming a diglyceride, and eventually forming a triglyceride. The stearate can also undergo a structural change to oleate, which can also undergo an acetyl transformation, forming the triglyceride. So how do we form this triglyceride? Well, the triglyceride is basically a covalent bond between the fatty acid chain, the palmitate, the stearate, or the oleate, at the carboxyl end of the fatty acid, the end of the molecule that has the COOH, with the hydroxyl end of the sugar in the glycerol molecule. So if we look at it, what we have is we have the glycerol with the 3 carbons linking up with the fatty acids, where we have the hydroxyl group binding to the binding to the hydrogen in the opposing hydroxyl group, where what we end up getting is 3 fatty acids attaching to the 3 carbons, forming the triglyceride. The triglyceride is not the only fat that can be formed in lipogenesis. We also have the phospholipid. Notice the big change in the triglyceride structure between the phospholipid and the triglyceride you just saw, where on the 3rd carbon, instead of linking with a 3rd hydrocarbon chain, a 3rd fatty acid, we have a phosphate that's going to link with the glycerol molecule. Or we have the steroid molecule. The steroid molecule is a 5-ringed fatty acid with an R side chain. The R side chain will vary based off of the type of cholesterol that we're looking at, or the steroid-derived or the stanol hormone that comes from it. Cholesterol is necessary for stabilization of the cell membrane. The phospholipid is important for development of the cell membrane.